morning, class. We're going to skip the syllabus today. We're going to go right into all the work. I am Dr. Call, and today we're going to be learning about the cytoskeleton, but more importantly, the history of the cytoskeleton. In 1903, Russian cytologist Nikolai K. Koltsov hypothesized that an intercellular network of tubules determined the shape of cells. He deemed this tubular network the cytoskeleton. In 1942, there was a discovery by scientists that contraction can be reproduced in vitro by two proteins, actin and myosin, leading to our current understanding of the cytoskeletal role in muscle biochemistry. The cytoskeleton was thought to exist only in eukaryotes until the 90s. In 1992, when it was found to be present in prokaryotes, research revealed that the cytoskeleton actually originated in prokaryotes with the discovery of bacterial proteins homologous to eukaryote tubulin and actin. We now know the cytoskeleton to be present in both prokaryotic and eukaryotic cell types.
So I understand the historical significance and all that, but what exactly is the role of the cytoskeleton?
Oh, that's simple. And that's it. That's the cytoskeleton. Are there any questions?
I'm really confused. Can you say that again?
Okay.
Actually, how about I tell you all a story instead of trying to draw this out? I'm going to tell you the story about a dog named Kinnison and his dog friend, Lord Dognane.
Yo, buddy, you still living near the new clay house?
Yeah, dog, what's up?
You want to bring me some of those cool new treats that Endo Dogmic Retell You Yum makes? I heard they make it all weird and hyphy.
Yeah, dog, I got you. You the homie. I'll be right there.
Awesome. Thanks, buddy. Just be careful of the doji apparatus. I hear there's been a lot of heated arguments between the cisterns and the vesiculars in that part of town.
No worries, dog. I have some homies up in the cistern.
Cool, buddy. See you soon.
I'll be there. Lickety split.
Kinnison leaves the endodogmat retalium and heads to the doji apparatus. So you see Kinnison's at the kinesin walking across the road or the microtubules towards the positive end, or in this case, Lord Dognane.
Wait, so the microtubules are like roads?
Exactly!
Wait, so what did you mean by plus end?
Filamentous actin is polar, and therefore the two ends of the filament are different. Because of this polarity, the filaments often fit into other cellular structures in only one orientation, in a similar manner to how many parts of man-made structures are assembled. Growth, the addition of monomeric actin to the filament, occurs predominantly in one direction, the plus direction. This end of the filament is known as the barbed end, and the other end is called the pointed end, or the minus end. An actin-binding protein, coflin, is responsible for depolymerization at the minus end of filaments, which inhibits reassembly. It severs actin filaments by creating more positive ends on the filament fragments. These monomers can be recycled by profilin, which activates monomers to go back into the filament form again by an ADP to ATP exchange. The newly formed ATP actin is ready for assembly. Anyways, let's get back to the story. Kinnison continues down the road when he encounters a dangerous obstacle. Anxious about Lord Dognane waiting for his bone, Kinnison knows he has to do something quickly to get past the cat to complete his important delivery. Kinnison uses muscle contraction to get the strength and movement to fight off the cat, and then...
Whoa. Pump your brakes, Mr. Kolb. How exactly is it that the microfilaments are involved in muscle movement?
Muscle contractions involve actin filaments, which are thin, and myosin filaments, which are thick.
Tropomyosin blocks the active sites on the actin filaments where the myosin heads bind. When calcium ions are released from the sarcoplasmic reticulum, they bind to troponin, causing a conformational change in the tropomyosin, which then exposes the active sites on the actin filaments. The myosin heads then bind to the active sites on the actin and pull the actin filament along, shortening the muscle length and causing contraction.
This seems like a lot of work. Where does myosin get the energy to move the actin filament?
Another great question.
The energy from this process comes from ATP. An ATP molecule attaches to the myosin head. ATP then becomes hydrolyzed, and the energy released is used to cause a conformational change in the myosin head, moving the head and the actin filament along with it.
All right, now where was I? Oh yeah.
Oh, look, a dog.
Oh man, another annoying human, all obsessed with me. How am I gonna get past her? I can just use the power of the microfilaments to divide her in half and get right through.
Whoa. How do you split someone in half? We can't really split cells in half, can we?
Pretty crazy, right? We've actually been dividing ourselves since we were in the womb. It's an amazing process, and it's actually another great example of the importance of the cytoskeleton.
Microtubule growth begins at the centrosome matrix, which contains two centrioles. This is the negative end of the microtubules. 3 different types of microtubules are created: kinetochore, polar, and astrial. The kinetochore microtubules attach their positive ends to the centromeres of the chromosomes located within the cell. Astrial microtubules develop in the actin skeleton and interact with the cell cortex to aid spindle orientation. Polar microtubules interdigitate at spindle midzone and push the spindle poles apart via motor proteins.
All right, let's see if Kinney-san is successful in splitting the human in half.
Finally, after a rigorous expedition, Kinney-san arrives at Lord Dogning's crib where he's hosting a poppin' party.
Yo, Kenny-san, you got the stuff I asked for?
I sure do. Man, I wouldn't let you down.
What kind of trash is this? I wanted the good stuff. This is a knockoff bone.
Ayo, I was told this was the best of the best. I don't know what to tell you.
Forget it. I'll go get it myself. No one can do anything right around here.
So why can't Kenny-san just go back to get the right bone?
Well, remember, Kinney-san represents the kinesin, which can only move towards the plus end from the direction of the nucleus to the plasma membrane. Lord Dogning represents the dynein, which walks in the opposite direction. But remember that it takes energy for motor proteins to move. Just like Kinney-san and Lord Dogning require more energy in the form of treats to walk, motor proteins instead use ATP. As one part of the motor head binds to the beta tubulin of the microtubule, it releases ADP, which frees up a spot for ATP to bind. Then a conformational change occurs, causing the other part of the motor head to take a step forward while the head attached to the ADP stays in place. The head behind now hydrolyzes ATP, releasing a phosphate, while the head in front binds to the beta tubulin, releases ADP, and repeats the process.
Wow, that was an amazing story.
Lord Dogning is kind of a jerk, though.
Well, if Kenny-san wasn't so irresponsible.
Hey now, you're missing the whole point of the story.
Dr. Kalb was right. The important thing is that we now know how the cytoskeleton works.
Who needs textbooks when you have dogs?